I'm a professor in the plant sciences department here at Montana State University. If you've opted to just watch the videos online, I've uploaded 2 shorter videos about my research in honeybees, as well as our efforts involved in SARS-CoV-2 testing at the local hospital in collaboration with the MSU research project. So those recordings might be a little bit higher quality than this one today. So if you've just opted to listen online, that might be your best bet. But for those of you in the room now, and for those of you opting for this version of the live version, I'm going to give a short presentation today and then leave plenty of time for questions and answers, which I think is the main goal of the classroom component of this course. So again, the focus of my research here at Montana State University is investigating the impact of pathogens, but primarily viruses, on agriculturally important systems. The system I'll talk about today and is the primary research subject in my lab is honeybees. But we also have a project on investigating the impact of a potato virus Y, on seed potato plants in Montana and beyond. So the first question you may have is why investigate the impacts of pathogens on honeybees? And it's not only because they produce honey, but bees are important in agriculture because they pollinate many of the crops that produce the fruits, nuts, and vegetables we enjoy. And without honeybee pollinators, the shelves on your stores would look drastically different and more like the image on the right. So bees are important plant pollinators. They collect pollen as their protein source, and you can see some bees here on the left with pollen stuffed in their pollen baskets. They'll store that in the hive in their colony for their protein throughout the winter months. One of the clearest examples of our reliance on honeybee pollinators is the almond pollination event, which occurs each February in the Central Valley of California. And for this single pollination event, which again is the world's largest, over 1.8 million colonies of bees have to be transported on flatbed trucks, typically, to the Central Valley of California. And every box or colony of bees has about 40,000 bees in the box. So I'll let you do the math. 1.8 million times 40,000 is how many bees there are in the Central Valley of California each February. So you may not appreciate that Montana is also a big honeybee keeping state. We provide over 150,000 colonies that are moved from Montana to California around Thanksgiving time each year to aid in this pollination event. So I hope that you've heard by now that unfortunately honeybees and other native and wild bees have suffered unsustainable losses. For honeybees, that annual loss have averaged 33% annually since 2006. There are many factors involved in addressing this question, what's killing the bees? We appreciate that multiple factors are involved, and those include agrochemical exposure, inadequate floral resources or nutrition, and pathogens, which is again the primary aspect of what we study in my lab. In early days, it was appreciated that pathogens were more prevalent in CCD, or colonies affected by colony collapse disorder, which is only a minor percentage of the reason why colonies die, compared to healthy colonies. So this was kind of our initial clue that pathogens may play a role in colony losses. And so we and others hypothesized this. And this is a real epidemiologic challenge. I think we can relate with the importance of epidemiology and studying disease throughout populations and transmission. We don't have a CDC for honeybees, and so my lab and others are forming this kind of coalition to figure out what is the role of pathogens on these colony deaths. So pathogens, I've mentioned a lot about them already. For honeybees, they include the same type of categories of pathogens that infect you and I. That includes bacterial pathogens, fungal pathogens, eukaryotic pathogens, such as protozoans and trypanosomatids. And then the largest class of honeybee-infected microbial pathogens are viruses, and viruses of a particular category with RNA genomes. So we call them RNA viruses. They have positive sense RNA genomes. That means they're mRNA-like and ribosome-ready. So these RNA viruses are the largest category of bee-infecting pathogens. What you're looking at here on the top is a transmission electron microscope image of one virus that's called Lake Sinai virus 2. It's really small. It looks like a little soccer ball, about 30 nanometers in diameter, meaning about 10,000 of them would fit across the diameter of a single human hair. And that's why we need the transmission electron microscope rather than a light microscope. You can't see most viruses on a light microscope to image this virus. In the lower panel here, you see a bee with an ectoparasitic mite called Varroa destructor walking on her back there. And this mite is not only one of the most detrimental pathogens to honeybees in its own right, it drinks the blood or hemolymph of bees and as well as developing larvae and kills them, but it also serves to transmit many of these RNA viruses. So not unlike mosquito and ticks, transmit viruses between different humans or different animals like birds and people or birds and wildlife. This mite transmits honeybee viruses around and between colonies. So the focus of my research again is how do viruses impact bee health at the colony level, so the superorganism level, all 40,000 bees in the box, as well as the individual or organismal level. We also do experiments at the cellular and molecular level. We do that by carrying out field and colony level monitoring studies. We team up with commercial beekeeping operations for those studies that manage thousands of colonies. And in laboratory-based experiments where we bring bees into the lab and culture primary honeybee cells as well. So when I think about the honeybee immune system or the antiviral defense mechanism, this is what I think about at the individual level. You have a bee here. Of course, the mite impacts the virus outcome because it transmits particular strains of the viruses maybe. At the cellular level, we have molecular mechanisms that honeybees, just like you and I, have evolved to defend themselves against viral infection. And one of these key antiviral defense mechanisms in insects and plants, which I want you to look up and read about later, is called RNA interference. And for those of you who it's been a while that we have had genetics, RNA interference just does what its name suggests, and we'll go over that in some detail in the upcoming slides. So again, to investigate the mechanisms, we actually do experiments in the lab. The upper left-hand corner here shows a frame of bees. These are newly emerging adult bees. You can see their little heads sticking out here. They chew their way out of these cells that look like honeycomb, but instead of having honey stored inside, that's where the babies develop into adults. It takes about 21 days for that. In the middle image here on the top, you see Finale Perrick. She's a graduate student in the lab. She's giving the bees an artificial mite bite with a dose of virus and then double-stranded RNA, which we appreciate is an immunostimulatory molecule. And then the bees live out their days in these little deli-style apartments in the lab. And then on the lower panel, you see larvae. We harvest bee larvae and then obtain cells from them. These are what we call primary cells, meaning they won't divide forever in culture conditions. But you see on the lower right, hemocytes, which are the immune cells of honeybees, growing in vitro in a cell culture dish in the lab. And we use these systems to investigate molecular mechanisms of antiviral defense in the lab. Importantly, we use what's called model viruses. Those include mosquito and human infecting viruses, the Symbis GFP, that's tagged with green fluorescent protein. Flockhouse virus is another commonly studied insect virus that we use. And then we also use bee viruses, including deform wing virus and Lake Sinai virus too. So I mentioned model viruses. One of them is this green glowing virus that's been tagged with green fluorescent protein. You can see on the bottom here, this experimental setup. This is Laura Brucher, who has her PhD from my lab a couple years ago now. She's injecting bees with either virus alone or virus plus double-stranded RNA. The bees live out the duration of this experiment in these deli-style apartments. And then we monitor virus production as by many different ways. One is qualitatively by fluorescence microscopy. You see the green there, and that's due to the glowing virus that's infecting the bee. We can also do a technique called Western blotting, which shows us how much protein or viral protein is in the honeybee. And then primarily we use quantitative PCR, which allows us to look at the number of copies of RNA, that's genomic RNA as well as transcripts that the bee has, as an indicator of how much virus the bee is harboring. So I mentioned RNA interference. As students, I think it's a good thing for you to look up later. It is a post-transcriptional gene silencing mechanism. So just a little review of the central dogma. So our genomes are composed of double-stranded DNA as well as honeybees, which are transcribed into RNA, and then those RNAs are spliced and exported from the nucleus and into the cytosol, where those mature messenger RNAs are then translated by ribosomes into proteins, which we generally consider are the functional unit of life. So RNA interference, again, does just what its name suggests. The honeybee and you and I actually encode proteins that target that RNA and target it for destruction. And if the RNA is destroyed, you don't get any protein production. And we think about a virus, you can think about a virus as a protein shell encapsulating its genome. And without the protein shell, you don't get any more production of any virus. So no protein, no virus. That's why RNA interference is an effective antiviral mechanism in plants and insects. And it's a gene regulatory mechanism in you and I. So I just want to give you a snapshot of the data from Laura's experiments. So I mentioned that she injected the green virus in the presence or absence of double-stranded RNA. I wanted to emphasize on this RNAi slide that it's a sequence-specific mechanism. And so an RNA is used to target the cognate or same RNA for destruction. And so when we inject specific RNA to the virus, so it matches the RNA to the virus, we get reduction of the virus. In the case of only RNAi functioning, you would not expect then that the controls, which we inject non-specific double-stranded RNA, that's the same sequence as a luciferase gene, for example, to reduce the viral abundance as well. And so what we found here is that we found that both specific double-stranded RNA and non-specific double-stranded RNA reduce virus in the honeybee, indicating that it's not all about RNAi in the bee. RNAi does function to reduce virus, but in honeybees, non-specific double-stranded RNA also results in the downregulation of viral production. This is more similar actually to you and I. If I injected, and I won't, don't worry, double-stranded RNA into either any of you right now, you would have what's called an interferon response. It's a general antiviral response to reduce viral infection. So it seems like honeybees have both a general antiviral response as well as a specific RNAi-mediated response. When you think about how colonies live, I don't have a picture of that next, but they live in very crowded conditions. I mentioned of over 40,000 individuals. So it kind of makes sense that honeybees need a general, fast, non-specific antiviral response to combat viral infection. But what is that? And so to delve into that further, what we did is we took individual bees from those experiments, multiple experiments. You always have to do lots of experiments in science. We screened them for pre-existing infections. We selected those that had primarily the virus infection we were studying. We sequenced representative individuals, and that means we sequenced all the RNA that was in each individual bee, and that's called transcriptomic analysis, or RNA-seq. And it gives you a snapshot of the genes that are being expressed or turned on at any given point in time. And in our case, it was in response to virus infection or double-stranded RNA only. So we wanted to look at those two cases as well as those cases in combination and compare them, of course, to a control, which was just mock infected and injected with saline. So I'm not going to go over the details of that, but the honeybee encodes over 16,000 genes. We found that hundreds of them were differentially regulated, and a lot of them exhibited increased expression. I've shown some of those genes in the context of a cell in green on this slide. And so when we as scientists are trying to figure out from these hundreds of genes which ones are more important, we place them in with their known roles based on what we know of the gene function in those in bees as well as in Drosophila, which is a model insect. It's the fruit fly. And we kind of make hypotheses about which pathways are important for antiviral defense. And in doing that, we figured out that the RNAi system is indeed important. We also discovered completely uncharacterized genes. That's why one of them is called MF116383, listed at the bottom of the slide. because it's just given an arbitrary name on the NCBI, that database of genes that's freely accessible to all, and as well as the heat shock protein response. And we've determined and gone on to do studies that all of these genes and pathways are important in honeybee antiviral defense. So at this point, you think, wow, she really gets into the details of the mechanisms of honeybee antiviral defense, and we maybe you've lost sight of why we care. Again, why we care is that bees are dying, and one of the ways that you could imagine you could fight off or you could prevent bees dying from viruses is you could have bees that were more well suited, to fend off viral infections. And so the long-term implications of identifying the genes that are important for honeybee antiviral defense is we can team them up with other scientists. And in the lower right-hand corner is Dr. Sue Kobe. She's at Washington State University. She's actually a honeybee breeder. She breeds honeybees for different traits such as gentleness, honey production, propolis production, and things like that. And so if we can give her a list of genes that honeybees have naturally evolved on their own to combat viral infection, she can include them in her selective breeding programs in the future. So this is down the road still, but this would help beekeepers in the end have colonies that were just naturally better able to withstand viral infection on their own, rather than introducing the next magic pill for bees to fend off their viral infection. So that's primarily what we do in the lab. I wanted to show some pictures of the people that actually do the work. I would usually say, to me, they don't look very much different from those of you in the audience. Today, I unfortunately can't see you, but I want you to be able to place yourself and think of yourself as potentially a future scientist or someone who reads about science. And all the work that's done in my lab is carried out by undergraduate and graduate students, as well as research technicians, including Dr. Katie Doffenbaugh, which is pictured here. Importantly, a large part of my job is getting funding for this research. So I write lots of grants. So this research is your and your parents and your friends' taxpayer dollars at work. And this funding comes from the National Science Foundation, the USDA or Department of Agriculture, the Montana Department of Agriculture, as well as private foundations such as Project APIS-M that fund honeybee research. And without this funding, we couldn't address important problems such as what's killing the bees. That's not something that non-academic researchers would be spending very much time looking into. Although now I hope that you appreciate that it's a fundamental component of agricultural that's often overlooked. So in the last couple minutes, I wanted to alert you to some videos that you can look at online. They'll talk about the mechanisms of what we do. I also want to let you know that I teach general genetics. I'll be teaching it this spring, currently teaching virology, which of course is an important topic now. Both an undergrad and graduate section of virology that happens every fall. And then I just want to take a few minutes to tell you how we transferred our expertise in RNA viruses that infect honeybees to RNA viruses that infect humans. And I think you all appreciate now that SARS-CoV-2 is a positive sense single-stranded RNA virus, so the same category of viruses that I study that infects bees. Of course, SARS-CoV-2 doesn't infect bees, and those bee viruses I told you about don't infect humans. SARS-CoV-2 is, of course, the causative agent of the disease known as COVID-19. And early days, back in March, when Bozeman was first being faced with this, I and others at Montana State University teamed up with partners at Bozeman Health Deaconess Hospital to address the situation and start testing SARS-CoV-2 in patient samples at Bozeman Health. So you can read more about these activities on recent news articles. But I wanted to give you, the biotech class, a bit of a sense. Many of you probably will have SARS-CoV testing, and I wanted to talk about this for a bit today. The testing that we developed, and well, we didn't develop, we just utilized. The test was developed by other scientists before us, but it was a qPCR technique. So that same technique we use to detect nucleic acid of viruses that infect bees is what's used to detect the nucleic acid of the viruses that infect humans. It's a sequence-specific amplification, and so what you're doing is looking for specifically SARS-CoV-2 genomes in patient samples. That's a nucleic acid-based test. There are also, and we'll hear more about in the months to come, antibody-based tests. Those can either be of two flavors. One is to determine, do you have any antibodies that are recognize SARS-CoV-2, meaning have you had a prior exposure to SARS-CoV-2? and your B cells, B lymphocyte cells, develop antibodies, and those antibodies find and bind SARS-CoV-2. Another way antibodies are used is like by biotech people and scientists like me, is that we use antibodies to actually detect the virus that's circulating. So a patient sample that had SARS-CoV in it would have antibodies. They might be bound to a strip, not unlike a strep throat test or a pregnancy test. Those are antibodies that are bound to that matrix. And those antibodies recognize SARS. A second antibody is also there. And a colorimetric assay is used to detect the virus. Again, that's protein or a virus or antibody-based testing. What we did is nucleic acid-based testing. I don't need to remind you again that nucleic acids are DNA and RNA, and the SARS-CoV-2 genome is RNA. How do we detect them? Well, I showed you a transmission electron microscope image earlier. This wouldn't be a great idea for SARS or any of the viruses that we study, because a lot of viruses under the transmission microscope look similar. These are black queen cell virus, deformed wing virus, which infect bees, and then the polio virus, which of course infects humans. And all of these are 30 nanometer icosahedral or soccer ball shaped viruses. So you're not going to use a microscope. There are other detection mechanisms, such as growing them in culture and plaque assays. There are serology or antibody mediated tests, and then there are nucleic acid based tests, such as polymerase chain reaction. So we use the PCR-based test to detect the RNA. I'm going to click through these slides pretty quickly. The RNA of this course of SARS is housed inside the virion. It's pictured here, and typically it's covered by this protein N called nucleocapsid protein. But importantly, you see there's a lot of N in there. There's a lot of green balls here. The gene that encodes nucleocapsid protein, that's encoded by the SARS-CoV-2 virus, that's the RNA we detect when we're looking for it. So we're detecting a very abundant RNA species when we're looking at SARS. The first step in that is to get a patient sample from the hospital, of course, under proper biosafety protection and in a biosafety hood. We extract RNA from the patient sample, whether that's a nasopharyngeal swab or some other sample. We extract the RNA. We do what's called a reverse transcription step to make that RNA into cDNA. This process is described in those videos I mentioned earlier that you can check out. And then we use PCR, quantitative PCR, to detect which RNA, if the SARS-CoV-2 RNA is there. Now, you might be hearing in the news that false negatives and false positive tests. I will say that the qPCR-based test, that's the gold standard for SARS-CoV testing, is rarely going to give you false positives or false negatives unless the sample, the swab, or the patient sample that was collected was inadequate. So this qPCR test is very sensitive and accurate, and it's what's called a TACMAN PCR assay. And if you look to the right here and Google TACMAN PCR, there's great videos online about it. But basically, for the PCR reaction, you need not only the PCR reaction to work using specific primers, but you'll see in the middle there's a probe. And that probe needs to bind that middle section of your target. Remember, our target in this case was the N gene that's encoded by the SARS-CoV-2 genome. And that probe has to be bound, and then the polymerase lifts and chews up that probe, and a fluorescent signature is released. So there's double specificity to ensure accuracy with this qPCR-based testing for SARS-CoV-2. Again, I went over that pretty fast, and there are just cool videos that you can look at online. One is called Honeybee Pathogen Detection, that kind of goes over these steps in more detail, and you can check that out. And again, we use the same techniques that we use to detect honeybee viruses in honeybee samples are the same techniques we use to detect SARS-CoV-2 from patient samples. So at this point, I'll end there and open it up for questions.
We're getting there. Hang on.
So, whoa, that's odd. This is going to be a little controversial, but why not change the plant and then cut out the middleman of the bee? Why save the bee at all when we can, adjust the plant?
Oh, okay. So bees, pollinators, pollinate lots of different plants, like numerous crop species, but as well as native and wild plants that are important for the biodiversity of the planet in general. Sure, that's not a bad suggestion. And they're looking at trying to have, you know, breed crops that don't need honeybee pollinators, such as almond trees, for example. They haven't done it yet, but there are certainly crops that don't require pollinators. Seedless mandarins is an example. Bananas is another example. When you breed crops that are seedless and no longer result on that natural mating process, you lose diversity in that crop in general. And if you, I'll use bananas as an example. They had a specific banana strain, I believe it was called Big Mike, and it was the kind of bananas that everybody was eating. That strain of banana became susceptible to a certain fungal disease, decimated all banana plants. And as if you think about biting into a banana, you no longer have seeds. So you no longer have anything to work with. And so you go down that evolutionary path where you don't need pollinators and you don't have natural mating anymore between plant species. You're going to lose hybrid vigor in general, and you're going to go down a dangerous evolutionary path. You can do it, sure, for some almond groves, but imagine if every almond grove was exactly the same, how susceptible they would be to a new disease. And I think, as we can appreciate now with the SARS-CoV-2 pandemic, it's good that we have variations in our ability to defend ourselves against that virus. And if we were all as highly susceptible, that would be bad for any population of any wild organism. So it's not a bad idea, and definitely plant breeders, that's one way to go across it. The reduction in pollinators is to develop plants that don't need pollination, but you're not going to be able to do that for every crop, nor the other species that require pollinators.
Here's another question. I'll ask one while I'm walking over there.
Okay.
Humans have a lot of retroviruses in their DNA. Do bees also have the same?
Yeah, that's a good question, and one we don't really know as much. The bee genome is less well characterized and investigated. There have been evidence of RNA viruses actually integrated into the bee genome that don't encode a reverse transcriptase, so they're not necessarily retroviruses. Most eukaryotic organisms have relics of retroviruses, and they could be now evolved into more like transposon sequences in their genomes. So I would guess honeybees do, but they're just undercharacterized, and there is no example of a retrovirus that infects honeybees that's been detected to date, but that doesn't mean it's not out there.
Another question.
So what is it about the past couple decades or a few decades that has kind of led to this big decline in bee populations? Has there just been like a more unusually like rapid development in bee viruses?
No, so I tried to introduce this at the top of the lecture, but it's appreciated that multiple factors are likely involved. And a lot of problems that are really important and interesting, it's not going to be just one thing that's right. It'd be easy to fix if it was one agrochemical, for example, or one class of agrochemical. We could ban it and then be done with that problem. So to date, it's appreciated that multiple factors are likely involved. Agrochemical use, and even there could be sublethal effects of agrochemicals, not just acute deaths. I mean, that's easy to figure out because there'll just be a bunch of dead bees, but there could be sublethal effects. There's also inadequate floral resources. So when you think about it, just the management of our environment, if we don't let flowers in the ditches in the roadway, for example, come to bloom, if those are mowed before bloom, or if alfalfa is cut before it blooms, then bees lose out on that nutritional opportunities. The more, for native and wild bees, that one of the biggest effects and impacts is paving things and just in general development, because then there's less habitat for those ground nesting bees. So bumblebees and a lot of native and wild bee species nest in the ground, and habitat destruction is probably one of the biggest causes of losses there of those bees. And for honeybees, it's unknown why. Pathogens are just one factor, and it's what I study, so it was the focus of the talk today. But probably, you know, we haven't found one virus that always is associated with all colony deaths. And so you can also appreciate that often at any given point in time, a honeybee colony maybe has several different types of infections going on at the same time. And so we need to figure out what are the most detrimental combinations of all these factors that are bad for bees, and are there management strategies that we can do? So beekeepers, for example, treat against mites. They use miticides, which can, you know, then the question becomes, how bad are those miticides for bees? Of course, they've been tested to the extent that they can be. And so they treat the bees with miticides so that you kill the mites, which would kill your colony. And then they reduce viral transmission. So your virus load of mite transmitted viruses should be down. But again, I hope that this long answer is giving you the idea that there are multiple factors. And even so, that when one colony dies, why did it particularly die? Unless it's an overt bacterial infection that you can smell, or heavy mite infestation, which you can easily see, or acute pesticide or insecticide exposure that, again, you'll see a bunch of dead bees. Often it's a combination of factors, and trying to understand why now over the last 10 years has this been such a big issue is a big open question. You know, it also brings up the topic of understanding large scale monoagriculture, which is what bees participate in. You know, that's a bigger system than ever now, but that's not something that just happened in the last 10 years as well. But it's something to consider. You can't pollinate the almond groves with native and wild bees in California. There aren't enough of them during February at that time of year, and those almond groves are big. Honeybees are one of the insects that can fly like 2 miles from their home in a radius. So they can penetrate large-scale mono-agricultural fields and crops like almonds better than other native and wild bees can, that they would just forage on the edge. So yeah, not a great answer, but that's why it's still kind of a mystery. How are all these factors coming together such that bees are suffering high annual losses? I do want to mention one last thing, is that beekeepers in the US have been doing a phenomenal job addressing this problem. So I mentioned that 30%, imagine you're a beekeeper and you go out to your bee yard and 30% of your bees are dead, your colonies are dead. What beekeepers can do is they do a thing called splitting the colony. They'll have an empty box here, they'll have a bigger box here, and they'll move several frames of bees into this new box, and they'll either introduce a queen bee or transfer a queen cell to that new box. And then now they have 2 colonies from one. So with 30% losses, beekeepers can make up for those losses, and they have every year, to maintain our pollination force to about 2 million to 2.5 million colonies nationally. But without beekeepers doing extra work, we would have an insufficient number of managed honeybee colonies for pollination. So beekeepers have really stepped up to this challenge.
So let's see, we have one back here. I'll have a question. Bats and bees and humans are all social animals. What can you make from that? Not all animals are so social. Could you explain something about that?
Yeah, so it's really interesting because bees in particular, they, if you think of this order, insect order called Hymenoptera, you have completely what we call eusocial insects. So one honeybee can't live on its own. It'll die without the colony. And so that's a eusocial insects. Ants are like that too, where there's a queen and then there are workers and they have different tasks. Those tasks include foraging for food, rearing the young, defending the colony, as well as laying the eggs. The reproductive unit of the colony is the queen. And so they've really established and depend upon this colony. Where there are other bees like bumblebees and other bees that are such as leafcutter bees that are solitary. And then bumblebees are a curious case because they're solitary in the spring. There's only one queen bee that overwinters. And then she creates a colony of bees in the hundreds during her life. So it's really interesting to study, the different genetics of what's called sociality, or from solitary to eusocial. And then are there consequences in, what we study and care about is the immune system. So you can imagine that a little Drosophila, that's a solitary fruit fly, has to have an all-encompassing immune system to fend off all pathogens. Where you could envision that honeybees, maybe they have different immune mechanisms. They talk about a propolis envelope that's been created around the colony. They talk about behavioral fever, where the bees actually crank up the temperature inside their colony. We don't know if these are really reducing any kind of pathogen, but it's interesting to speculate on and think about. And so there are definitely genetic consequences that beyond their behavior likely have other impacts and maybe even on their immune systems too.
So how do viral infections spread between bees? Is it similar to humans? Is it through shared fluids or food sources?
Yeah, that's a very good question. So one thing this brings up too is, so we call it vertical transmission when it's from mother or father to baby bee. That's vertical transmission. We call it horizontal transmission when it's half sister to half sister, and they do a thing called feeding, mouth-to-mouth feeding, called trophallaxis. When one bee goes and drinks some nectar from a flower, she comes back to the colony. She does actually mouth-to-mouth feeding to feed it to a house bee, who then goes and stores that nectar and dries it down for honey. So that's horizontal transmission. Bee viruses have also been documented to be transmitted on flowers, so surfaces, not unlike the door handle. And actually, sexual transmission of bee viruses happens too. Bee viruses have been detected in the semen from males, and so they can be sexually transmitted just like human viruses as well. So all the routes that you think of, as well as vector mediated. We talked about mite mediated transmission. So all those typical infection routes work in the bee colony as well. Now think about, though, when we're in this era of physical distancing, otherwise known as social distancing, that we're in now, that's not possible in a honeybee colony. There are 40,000 individuals crawling all over each other. And so it's a perfect way for horizontal transmission of viruses in a bee colony.
We're looking for another question. I've been on this campus quite a while, and I find that Michelle is one of the most active researchers here. And as I said earlier in the course, the best thing you can do is put your right foot in the door of a lab like Michelle's and get research experience. What we have found in the past 10 years, by developing a university that is research oriented, the students get a lot more out of it. You can only get so much from a talking head, even Michelle's. You have to get in the lab, wash dishes, and start, and pretty soon you'll find your own project. Are there any questions? So one thing we see in Michelle is that she can move from one field to another. She is not siloed in the way some scientists get to be, in that although she works on bees, she immediately responded to the COVID virus by equipping her lab in a way that could be used against the COVIDs. And so we're constantly looking at trying to avoid siloing where you can only do one thing in science. And the other thing is in course development. The one thing we've done in this course is to try to show you all the different silos, and then you can at least choose which one to specialize in a little bit. Are there any more questions? Sure, coming.
So I've seen a lot of news about neonicotinoids and trying to get legislation passed to ban those from use or heavily restrict them. Has that made a lot more, and there's been a lot of controversy around that, has that made more progress in California because so many of the cropping systems there rely on pollinators?
Yeah, so neonicotinoids are, of course, they're a class of insecticides, and they mess up the acetylcholine receptors in the insect, and then they paralyze them and cause death. They are insecticides. France banned them. I forget how many years ago now. So that's kind of a natural experiment in the works there. And I don't think it ended all of their bee problems. You know, there are still a lot of colonies that die in spite of a neonicotinoid ban. That said, there are exemptions to the ban. It's not 100% there in France, but they have been targeted in part because they've been used a lot more in the last 10 years. All insecticides kill bees. And so the idea is, I think the best idea when it's under the umbrella of integrated pest management is that the the goal to reduce the exposure of honeybees to insecticides is a goal that everybody should have. So for example, when these problems started to happen, people started to look at their practices a bit more, especially in California, which that's where I did my postdoc. So I started bee research in California, and they have a pesticide registry. So every drop of pesticide that's applied to California crops you can look that up online. And when I got to Montana, I looked for that same thing. And of course, we don't have that kind of thing. In California, then they could work with growers to say, hey, if you need honeybees to pollinate your crops. So especially almond growers got right on in there and actually fund some bee research, and also figured out that they were tank mixing certain fungicides and insecticides, and Reed Johnson at Ohio State University figured out this. They were just doing it to reduce workload, right? Spray once instead of twice. But they were spraying that during the day, and then so bees could interact with those blossoms during the day by not what they call doing tank mixing fungicides with insecticides any longer, and by spraying crops at night. So bees only fly when it's light. So if they spray at night, and then if they avoid spraying during the bloom, let's say they spray pre-bloom or post-bloom, if they can hold out, then bees won't interact with crops that have neonicotinoids on their blossoms at that time. And of course, like all chemicals, there's a certain half-life to those things. And presumably by reducing the exposure, you reduce the consequence. Now, unfortunately, some neonicotinoids, like for those that are used for seed coatings, for example, in the Midwest, it's hard for a farmer. It's nearly impossible, as I understand, if you're not doing strictly organic, to buy seeds that aren't just coated proactively in neonicotinoids. So we unfortunately have a lot of neonicotinoids out there. Now you can imagine if they have a longer half-life and they're in the ground and groundwater, that they could be taken up by the plants that bees forage on, and that, let's say around a cornfield, for example. There's been some evidence, but the data's mixed. How much neonicotinoid gets back into the nectar of those wild plants and floral species that grow along the outside? When they go out and do surveys for, systemic detection of pesticides, particularly insecticides, in non-target crops, so those are non-target plants, so those outside of the crops. So, sometimes you see high loads and sometimes you don't see high loads. And to what degree do these sublethal effects impair a bee colony? Those are really challenging questions to address. Of course, you can put bees in a little dish and expose them, and we get what's called an LD50 level. How much of this chemical, when directly applied to a bee, will kill it? But then that only gives you part of the story, right? How much sublethal dose would actually impair a colony's function? And those are harder questions to figure out. So we don't have a really good handle on it. One other thing to consider is that if you ban all neonicotinoids, which you could use less of than the organic compounds they used before, what are you asking farmers to go back to? If they're not organic, they'll go back to maybe something that was even worse for bees. So again, another important point, and something that needs really to be looked at in fine detail in different cropping systems. Of course, we also need to feed the world, and without some of these chemicals, you would have these root borers that just decimate crops, and we would be in food shortage issues. So it's, again, a complex problem that involves thinking about it holistically. Probably doesn't go to one extreme or the other, complete ban or just free for all. Some happy middle there with integrated pest management and reducing the amount of contact between bees and insecticides, which kill bees. is just really important. But you know, most people want to work in the positive, most, you know, parsimonious solution to this big problem. But it's a really important one that you raise.
We have another question back there, but I'll ask about killer bees, African killer bees. I believe our domestic normal bees are also an invasive species, right?
That's true. So honeybees, Apis mellifera is their scientific name. They were brought here with European settlers. So not unlike European settlers to North America are also an invasive species. So are honeybees. So the settlers brought them over to produce honey, and then now they're of course widely distributed in North America and South America, but they came over originally from Europe. Whereas things like bumblebees and leafcutter bees, we refer to as native and wild bees because many of them were in, at least in North America before, and maybe their ranges have expanded or changed or been reduced in the last couple hundred years or so. But definitely honeybees are a non-native species, but then for that matter, so are almond trees and almond groves, and a lot of the plants that we grow and have in the US are non-native species.
Where do the viral infections originate? For SARS-CoV-2, it's bats. Where do bee viruses come from?
So that's a good question. So really, this is kind of a interesting question. When you think about the start of life, really, you know, when we had this pre-biotic RNA world, probably viruses interacting or genomic parasites or nucleic acid parasites were probably co-evolved in the initial stages of life. And if you want to learn more about virus evolution, you can read papers by Eugene Koonin. That's Koonin, which are very interesting. So if you have a host cell and you have a virus, now what you're asking about is more immediate, like was there a spillover event? So viruses are obligate intracellular pathogens or parasites. They are in most hosts, most organisms have viruses. When we think about viruses, and we often think about them from a mammalian-centric way, we think about there are different viruses that infect, for example, our dog and cat versus ones that infect us. Or in the case of SARS-CoV, we think that in bats, are mammals too. They have, for reasons that's not completely understood, they can harbor and deal with a lot of viruses. SARS-CoV-2 is not killing a lot of bats, for example, and that's a pretty interesting research topic in its own right. But then when these mammalian viruses can switch hosts and that host has never experienced in them before, then often the new host is more detrimentally harmed from these virus introductions than it would have been. Now, some of these virus introductions don't go so far, right? If I, for example, got an influenza virus from a chicken farm because I worked at a chicken farm, but then I was pretty sick, but then it didn't pass on to my family members, I'm kind of what's called a dead end host. And that's what most of the spillover events end up being. Maybe somebody gets sick that was in immediate contact. The real problem is, and I know David Quammen and others have been here, the real problem comes in when those introduced viruses readily transmit from human to human, and now it's just a human virus. So different viruses have different host cell interactions. So SARS-CoV-2, for example, uses as one of its receptors the ACE2 receptor, and maybe our receptor in mammals is pretty conserved. So that virus then infects a bat, that infects a pangolin, that could infect then us. Maybe our receptors are just enough similar that that virus could still get in. And then once it does really well, it can kind of adapt in that new host or not. Likely it did. You know, the sequence from the bat is different than those sequences that are circulating in us right now, but not that different. So it could get into the cell and replicate and transmit. One important point that you brought up is like, so where do honeybee viruses come from? This is an important and general distinction. In plants and insects, the viruses are less host specific. For example, I think cucumber mosaic virus, for example, can infect over 200 different plant species or more. Like it's just, they get in through the plasmodesmata or the leaf surface, and there's not this receptor virus interaction. So they're less specific in the plant world. Likewise, in insect viruses, we call them honeybee viruses, but a lot of the viruses I study can infect ants, can infect bumblebees, can infect leafcutter bees, and they have a more what we call general or promiscuous interaction with the host. So where they came from, we don't know. We can do phylogenies or look at bees. Bees, like most organisms, came out of Africa initially when we had the giant landmass. And so we can see if something is deeply rooted on this genetic tree, it is likely co-evolved with bees for a really long time. And so it's hard to find out like where the viruses came from. At this point in time, many of them are just circulating in honeybee populations all across the globe. When bees were really dying in 2006, we stopped trade. So we used to be trading bees with Australia. Australia is an island. They can kind of maintain lower pathogen population potentially, and then we didn't want to bring in any new things. So as bees started to die more, we started to have more regulation on intercontinental transport of bees to reduce pathogen transmission, which I think is a very good thing. Before, nobody was thinking about that, right? Even now, bees aren't transmitted from Canada to the US. Only US queen bees can be shipped to Canada, but we're not getting colonies from Canada here. With the effort of trying to reduce some of that pathogen spread globally and intercontinentally. So a long answer to like bees co-evolved with their viruses, and where they come from, you could kind of tell if you bought an infected colony and then introduced it to a bee yard that had 200 other colonies, then this one colony would be the source. But the source for all viruses probably started with early days of eukaryotic life.
Thank you very much, Michelle. That was beautiful. Bye-bye.
Bye. Thanks.